Hi, everybody. It's Ms. Corcoran, and today we're going to talk about Chapter 21, Evolution of Populations. And as ever, I have loaded up here on the first slide the learning objectives set forth by the College Board. Let's take a second and talk about how there can be no evolution whatsoever unless there's variation. If everything were absolutely identical, then nature could not confer an advantage one way or another. No trait would be better than another trait, and evolution would not occur. So without variation, everything either persists or everything either dies off. So either everybody can compete or nobody can compete. There's no differential reproductive success. And if you have phenotypic variation or variation in your traits, it has to come from a change in the genetic code. So the question I'm posing to you right now is, where does variation come from? Two places. Mutation is the original source of variation. Straight up mistakes, changes in the DNA when it's replicating, or some sort of mistake during protein synthesis can result in a phenotypic change that can confer an advantage, or it can confer a disadvantage, and then that organism is dead. Another place that variation comes from, and most notably, it is from sexual reproduction. And if you think back to Bio 1 and that meiosis unit where we make egg and sperm, three things happen during sexual reproduction that increase variation. The first is the lining up of homologous chromosomes during prophase 1, where they wrap around each other and they actually share genetic information, and we call that crossing over. The second is when the homologous chromosomes line up in metaphase 1 of meiosis, and each pair lines up independently. We have 23 pairs of chromosomes for a total of 46. And I can assure you folks that there is no elf in each cell directing traffic and lining up homologous chromosomes saying everything from you got from your father on the left, everything you got from your mother on the right. Doesn't matter. Homologous pair number 1 lines up at the middle, completely independent from what homologous pair number 2 does, which is independent from what homologous pair number 3 does, and so on and so on and so on. So as you can see, a potential exists for a great deal of variation. And lastly, random fertilization is when a random sperm and a random egg get together after all of the potential variation happened during crossing over and independent assortment. And that's where the variation comes from in sexually reproducing organisms. If you don't reproduce sexually, if you're a single-celled organism, your main shot at variation is mutation. As we've said before, nature chooses. Nature says who's fit and who isn't. Natural selection acts on the individual, but populations as a whole evolve. That is in bright red. That is something you absolutely need to know. There's nothing an individual can do about their genetic makeup. They either have it or they don't. And this is also a review. Macroevolution are those large-scale changes we see in the phenotype of organisms, what we kind of think of when we think of evolution. And microevolution are those teeny tiny small genetic changes that lead then to changes in phenotype across a population. And I put an example down here, which is described as gene flow, which is just the movement of genes between different populations. So on the bottom left, we have a population of purple bugs. They are the same species as these brown bugs. They're just two different populations, and their genetic makeups are a little bit different. Let's say this little bug decides to go live over here with these bugs. He's bringing his genes with him, and he's going to change the genetic makeup of the population, or he's going to change the frequency of the genes in the population. So let's talk about that. If we look at this example of a field of flowers, this is the field of flowers at time 0. We've got red flowers, we have white flowers, and then the pink flowers are heterozygous. And we can see here that we have calculated, and don't worry about it, you're going to know how to do it by the end of the lesson. We've calculated the frequency of the dominant allele and the frequency of the recessive allele. And over time, we see in the next generation, the frequency has changed. And again, in the third generation, the frequency has changed. When we see over time a change in the gene frequency, when we see a change over time, What is that? That's evolution, folks. So here we're using numbers to actually prove that evolution is happening. I mean, you could take a look. You could say, well, gosh, there's white, pink, and red. And then by the third generation, there's only red. So something's happening and something is selecting for the red flowers or something's selecting against the white and the pink flowers. Which brings us to the first section, or excuse me, the second section in chapter 21. These two fellas, Hardy and Weinberg, they are mathematicians, and they got together and they talked about population genetics. And they decided to call the dominant allele P. Doesn't matter if it's big B for brown, little b for blue. If it's a dominant allele, they're just going to represent it with the variable P. They're going to call the recessive allele Q. Okay, doesn't matter if it's a little w, a little b, a little whatever, they're calling it Q. And they said if you add up all of the alleles in a population, then you'll have 100% of the, if you add up all the dominant and all the recessive, you're going to have 100% of the alleles in a population. And I think we can agree with that. So they came up with this fundamental statement of P plus Q equals 1. Okay, all the dominant alleles, all the recessive alleles equal 100% of the alleles. I think that's easy. And then they represented the genotypes. I know I'm throwing a lot of vocabulary from genetics back to you, and I'm going to go through it more slowly in just a second. But when they talk about genotypes, if you have the dominant allele and you're homozygous for that, then you've got PP or P squared. They said that's going to be the homozygous individuals. If you are homozygous recessive, Q being the recessive allele, then you're going to be QQ or Q squared. If you're heterozygous, then you've got a P and a Q. You could also have a Q and a P. So to account for the differences in the way you may have inherited your two different alleles, Mr. Hardy and Mr. Weinberg labeled the heterozygotes as 2PQ. The gene pool refers to all copies of allele at every locus and all members of a population. And once again, we represent that as P plus Q is 1. So basically their equation that they came up with is a mathematical tool that determines gene frequency in a hypothetical population that's not evolving at a given time. It must meet these 5 criteria below. And once we go through those, you'll be like, Ms. Corcoran, is that ever really happening? Is there ever really a population that's not experiencing those things? And the answer is really no. There's always some sort of pressure being exerted upon a population. But it can be applied to determine if the frequency of a gene is changing over time when we sample it at time A and we come back and sample at time B and we calculate those gene frequencies. And if we see a change, just like we did here, we can go, oh my gosh, evolution is happening. Now let's figure out why. Okay, so for a population to not be evolving, 5 criteria have to be met. First and foremost, the population has to be very, very large because small populations are subject to genetic drift, and that's something we'll talk about on Wednesday or in a couple days. There has to be no sexual selection. That means the mates can't be choosy. You could also refer to that as random mating has to be occurring. So you can either say no sexual selection or random mating's going on, but you cannot say no random mating because that infers very specific mating, which is some type of selection. So you can word that however you want, but make sure you word it correctly. There can be no natural selection acting on a population if we don't want evolution to occur. There can be no mutations, and there can be no gene flow. That means no purple bugs coming into the brown bug population and no brown bugs leaving and taking their genes with them. Okay. You can also refer to that as immigration I moving in or immigration E moving out. Notice the 2 stars by this. You need to be able to list the 5 criteria of Hardy Weinberg equilibrium. If those 5 criteria are met, that population's not evolving, but really and truly, that is a hypothetical scenario. So we're going to go through in just a minute. You're going to open the Google Doc, not right now, after the lesson I give you on my paper, and you're going to be completing the practice in groups, and then you'll open the key, and only when you've done it all, open the key and check your answers. Make a note of what you missed, and let me know if you don't understand it. So I'm going to pull up my Elmo, and I'm just going to write a couple things. This is where if we were in class together right now, I would be writing this stuff on the board. And I always say, if it's important enough for me to write on the board, you need to write it down as well. Okay, let's see. Zoom in a little bit more. All right, I'm going to try to use my very best handwriting. Just as a review, a gene. A gene is a segment of DNA that codes for a trait. Maybe in this case, the trait will be height. Okay. An allele is a different form of a gene. The trait is the same. The trait is height, but you could have big T, which is going to code for tall, or you could have little t, which codes for short. Both of those code for the trait of height. That looks sort of like a K. I'm making it a lowercase t. Well, my gosh, that's terrible. Excuse me. Okay. So let's draw our chromosome right here. Here's one chromosome. Here's another chromosome. Let's label that as a chromosome. And on that chromosome, we have a gene. This gene gives you information to make a tall organism. It will be big T. This information in the same locus or location has the information to make a short organism. That's a little t. So these would be two different alleles. If an individual inherited this pair of alleles from its parent, they would be heterozygous and their genotype would be big T, little t. All right, so here we go. There's the egg plus the sperm. They get together and they equal the first new cell of the new organism called the zygote. Okay, that's a Z. And that zygote has two alleles for every trait, only if they are sexually reproducing organisms. Okay. Obviously, if you don't use egg and sperm to reproduce, then you're not going to have two alleles for every trait. And you can have, if you have, keeping in our T's, if you are big T, you're homozygous dominant. And honestly, this is a review. If you had bio last year, this should be very familiar. But for you seniors, it's been a long time since you've had this. So please pay attention. If you have a big T and a little t, you are heterozygous. And if you have two little t's, then you are homozygous recessive. All right, the thing about this right here, these two, your homozygous dominant and your heterozygous, because you've got the dominant trait or the dominant allele, you're going to show the dominant phenotype. And remember, pheno means physical. I've run out of room, so I'm going to try to write phenotype. If you are homozygous recessive, you will show the recessive phenotype. So if I show you a picture of a tall plant, can you tell me definitively what the genotype of that plant is? You can't, because a tall plant could be homozygous for the genes, or they could be heterozygous. So you cannot tell. You can't tell the genotype. However, if I show you a short plant, you can take one look at that plant and say, Ms. Corcoran, the genotype of that obviously has to be little t, because it is a short plant. This is really important. You can always take a look at the recessive phenotype and know its genotype. And we will always start with that when we do these Hardy Weinberg problems. So I'm going to flip the page here. And review that Hardy Weinberg equation. I'm just going to put it as HW. They said P equals a dominant allele. Okay, we can be good with that. And that Q equals a recessive allele. Okay. And then they also said that if you add up all the dominant alleles and all the recessive alleles in a given population, you're going to have 100% of the alleles. So they came up with this statement that says P plus Q equals 1, or 1.0, or 100%. You must commit this to memory. This is very important. You're going to use this equation every single time you solve a Hardy Weinberg problem. This is alleles. Alleles. This has to do with DNA. Now, the other part of the Hardy Weinberg equation were the organisms and the genotypes. If you are homozygous dominant, here's your dominant allele. Homozygous dominant means you have 2 of those. Then it's P times P, or P squared. These are homozygous dominant individuals. Okay. And then over here, we're going to have our Q squared, which are homozygous recessive. Because if you have the recessive allele and you have 2 of them, it would be Q times Q, or Q squared. Then they said, well, what if they have 1 of each allele? You could inherit a P from your mom and a Q from your dad, or the Q from your mom and the P from your dad. And so to account for the different possibilities of the way you could inherit those alleles, they're going to put 2 times P times Q. So PQ is heterozygous, but because you can inherit them 2 different ways, they put a 2 in front of it. If you add up all of the homozygous genotypes plus all of the heterozygous genotypes and all of the homozygous recessive genotypes, then you're going to have 100% or 100% or 100% of the genotypes, which we're going to say is 1. You also need to memorize this. Okay, really important to know these 2 things. Once again, we will only ever be able to know the recessive genotype by looking at the recessive phenotype. Okay, and we'll get to that in just a second. Let's say you get a problem, and I want you to understand, when we do Hardy Weinberg, frequency equals percentage. Those words are going to be used interchangeably. So let's say big B codes for blue and little b codes for white in terms of flowers. And the frequency of the white allele is, let's see, is 0.9. What can we do with that? We've been told the frequency of an allele. It's the white allele, which means it's the recessive. And in Hardy Weinberg, the variable that stands for recessive is going to be Q. And what do we know? We know that P plus Q equals 1. So we've got P plus 0.9 equals 1. How do we solve for P? We can also write it as 1 minus 0.9 equals P, which means 0.1 equals P. So we used this equation, just like I told you. Ultimately, you want to find out what your P's and Q's are, because once you have your P's and Q's, you can actually plug it in to this equation. But you need to get to P and Q first. So let me give you another example. What if in a given population, of flowers, 36% are blue? These are the same examples. So big B equals blue, little b equals white. And if we remind ourselves what the Hardy Weinberg variables are, big B is going to stand for the P, and the little b is going to be the Q. Okay, so here's the trouble. We've been given the number of blue flowers. There are two possible genotypes if you have blue flowers. If you have blue flowers, you could be homozygous for your blue, or you could be heterozygous for your blue. And that just confuses things. We don't know what's what. So we really can't use this number to figure out what P is. We must always start with the one we know for a fact. So if out of 100% of flowers, 36 are blue, what percent is white? It's at 64. Okay, so 64% are white. I'm the worst at basic math. You're definitely going to need a calculator in this section. So 64% are white, which is little b, and from the Hardy Weinberg equation, it's also going to be Q squared. And I mentioned earlier that percentages are the exact same thing as frequencies. So we can get rid of this percentage and write it as a decimal, and it's the same exact thing. So 0.64 equals Q squared. Well, what do we do now? We actually have to figure out what Q is. If we can figure out what Q is, then we can figure out what P is, because remember, P plus Q, where is it? P plus Q is 1. Okay, so in order to solve for Q, we simply take the square root of each. The square root of 0.64 is 0.8 is Q. Now you're going to get really quick at doing this in your head when they're really good numbers. If it's a decimal, you can't do it in your head as quickly. But if Q is 0.8, and we remember that P plus Q equals 1, then what does that mean P must equal? 0.2. You got it. Now that we have P plus Q, we can answer any question they give us. So of the 36% blue flowers, the question may be what percent are homozygous dominant? Homozygous dominant would be big B, which equals P squared, which equals P is 0.2 times 0.2. So P squared would be, 0.2 times 0.2 is 0.04. Is that right? I don't have my phone handy to check my calculator. I feel quite certain you will tell me if it's wrong, but I think that's right. Okay. All right, so the next thing I'm going to have you guys do is a little bit of Hardy Weinberg practice on your own, and then we'll reconvene and go over those questions. I'll post the answers as well, so you can check them if you need to. But I hope that helped, and just let me know if you need any questions.